Okay, a very good morning, ladies and gentlemen, to all attendees, members of the faculty who are present, and the students. A very good morning again. So today we have one of the prominent researchers researcher of malaria, Professor Georges Snounou. So Prof. Georges Snounou's scientific career has been dedicated to the study of Plasmodium protozoan parasite, the causative agent of malaria. This parasitic disease, predominantly prevalent in the subtropical poorer areas of the globe, exacts immense health and economic burdens. His interest in these pathogens does not only rest on their major impact on public health, but also because they offer a quintessential model for fundamental biological studies. He actively pursued investigation that address four broad topics, mainly the genetic diversity of Plasmodium species that infect humans, the biology and immunology of the hepatic stage of the infection, the pathogenesis of cerebral malaria, and the molecular characterization of Plasmodium species in higher primates. So without further ado, Prof. George, you have 20 minutes to deliver your talk, and the screen is yours, Prof.
Thank you very much for your invitation. And I just would check that you see everything. Is everything okay? Can you see the screen?
Yeah, but I think it has to be in presentation mode, Prof.
I thought it was in presentation mode. How do I do that? It's
maybe you want to cancel this first.
Use slideshow. That's better?
Yeah, this
excellent. Anyway, thank you very much for your invitation. I'm very pleased to be participate in your webinar. And I had hoped to be in Malaysia for this webinar, but unfortunately, circumstances made it that I'm here in Paris, where it's -2 degrees and a little bit of snow, and I would have preferred to have been with you. Anyway, without further ado, let me talk to you today about an unexpected discovery, which is that there are two cryptic species in Plasmodium ovale. Now, I will go back to kind of historically how these things came about and why it is of interest. Plasmodium species in humans were discovered in 1880 by a French medic, Alphonse Lavran, who was working in Algeria. At the time was French. However, the studies of malaria was mainly done at that time by Italian scientists because, as you can see from the map here, Italy was a very highly malarious country, especially around the area of Rome. And this is where all the scientific research was done. A lot of the scientific research was done on malaria. And it is indeed these scientists who, in the last 10 years of the 19th century, have discovered that it was actually 3 species of malaria that existed, 2 of which caused the tertian fever, means that you get a fever peak once every other day. And then one of them, another species, Plasmodium malaria, has a quartan periodicity, which means that it comes every 3rd day. You get a peak of fever when the fever is going. And so these were named, these parasites, Plasmodium falciparum, Plasmodium vivax, Plasmodium malariae. And for the next 30 years, these were the only 3 species of malaria that were malaria parasites that were known. In 1922, a newcomer parasite appeared. John William Watson Stevens, working at the Liverpool School of Tropical Medicine and Hygiene, had observed in a soldier returning from West Africa. We don't exactly know where he was in West Africa, but probably Liberia, Sierra Leone, Nigeria. These were all British colonies at the time. On his return from duty to Liverpool, he was taken ill, and Stephens noticed that the parasites were strange. They were slightly different from P. vivax, and he's noted that the differences were sufficient for him to give them a name, Plasmodium ovale, in 1922. Ovale because some of the forms of the parasites, like the ones you see here and the ones you see there, have an oval shape. And this shape had never been seen in Plasmodium vivax, according to him, and therefore he gave it the name Plasmodium ovale. Now, this was a newcomer parasite that appeared, in a sense, 40 years after the discovery of Malaya parasites in general. It wasn't easily accepted. It's only until 1935 that people finally accepted that this is a different species. And as you can see, they are very difficult to distinguish morphologically. This is Plasmodium ovale, this is Plasmodium vivax, and I can assure you that if you look at it under the microscope, it is not easy if you see one or two forms only to see which one is which. Especially if you have very low parasitemias, and the course of infection of these two species tends to have relatively low parasitemias, around 5 to 10,000 parasites per microliter at peak infection. And therefore, there are not many parasites that are observed under the microscope. And if the parasitemia is low and you're looking at a thick SME, it is impossible to tell them apart. This is the reason why for many years, people did not accept that this was a different species, and they considered these to be variants of each other. But nonetheless, after 1935, and especially after experiments were done in humans to demonstrate that they were separate, that people accepted that Plasmodium ovale existed. Now, from 1935 onwards, and even from 1922 onwards, very little research has been done on Plasmodium ovale, mainly because the parasitemia, as I said, is low. Clinical severity is extremely rare. The patency, the time when you are actually ill, if you're not treated, is rather short. Within a couple of weeks, the parasites tend to disappear. It disappears by itself without treatment, and it's very sensitive to treatment at the time quinine, with just a few grains of quinine, and it'll disappear. And relapses, because it was a parasite that relapses, like Plasmodium vivax, are actually relatively rare. The prevalence is low, maybe because it is not noted, because there was a low parasitemias, and it was mainly observed in West Africa and very rarely in other parts of the world. And indeed, between 1922 and 1992, so this is over a period of 70 years after its discovery, there were only about 250 articles where people mentioned Plasmodium ovale. And I say only 250. This is out of about 39,000 articles published on malaria between these two in these 70 years. Of these 240 articles, only 40 were done by experimental infection of humans, which was possible during that time, especially in the context of malaria therapy, where malaria was used to treat neurosyphilis, and therefore observations of the course of infection were made. So as you can gather from this, very little is known about Plasmodium ovale and its course of infection and immunology, etc. Now, in the early 1990s, myself and others have with the sequencing of genes, we have exploited the differences that have been seen between genes, homologous genes between different species, and in particular the small subunit ribosomal RNA, which was sequenced for the 4 species of Plasmodium that infect humans. We noted that by comparing them, there are areas that are the blue areas here that are homologous, that are found in all the species, irrespective of what Plasmodium it is, and other areas that tend to be species specific. So we designed oligos, with PCR having been discovered in the mid 1980s, I'm sorry. We adapted a PCR, a nested PCR, in which we amplify one large part of the gene, and then we go specifically in a second PCR using the product from the first one to amplify using specific oligonucleotides another part, a smaller part of the gene. This is the nested PCR protocol, which is still being used today and is extremely sensitive in that one can detect one parasite in the sample. The genome, if there's one parasite genome in the sample, it will be detected by this method if it's properly applied. The first studies that were done with this gave an indication that actually the prevalence of P. ovale, which was considered to be extremely low in kind of 0.5, 0.1, if not 0.01% in many places, was actually underestimated because as people did studies on Plasmodium ovale in epidemiological settings using a sensitive amplification methodology, they found that the prevalence was actually higher than that, in the 5, 6, 7, 8, 9%, which is substantial considering the number of cases of malaria in the world. Now, during these studies, by ourselves and by others, we did get cases where we have a microscopic picture which is clearly a Plasmodium ovale, many times very typically Plasmodium ovale. However, when we did the PCR using the oligos that we and others has developed, it tended to be negative by PCR, despite the fact that there was enough DNA and the DNA was definitely positive for Plasmodium species, but negative for Plasmodium ovale, and of course negative for the other 3 species. Now, people who sequenced the genes from these funny samples found out that the small subunit ribosomal RNA genes of these parasites are different from these ovale parasites, are slightly different from the old Plasmodium ovale. I mean, that's why they called them a new Plasmodium ovale, as they found it. And indeed, when the sequence of the whole gene was done, it turns out that the oligos that I have been using to detect Plasmodium ovale, the ones in green here, were different by a little bit. And although that little bit of difference is sufficient to stop the PCR reactions from functioning correctly, they were different in the variant, what we called a variant ovale. And therefore, the term classic for the original sequence and variant became applied to these parasites, this new type of Plasmodium ovale, or this second population of Plasmodium ovale that was slightly different with respect to the small subunit ribosomal RNA. This was noticed in the 1990s, as you can see, the mid-1990s, and with time, because now that we can detect all the Plasmodium ovales, investigations of P. ovales has started to increase. And I can just I'm not going to go through all the epidemiological data, but mainly it was epidemiological studies in which PCR and molecular detection was included. As you can see, the number of articles published, for instance, between 2001 and 2010, a period of 10 years, was as much as what has been published in the previous 70 years between 1992 and 1993, 1922 and 1992. This increase of investigation epidemiologically was becoming more and more relevant, and as you can see, more articles, because with the wish to eliminate and fully control malaria and eliminate it, if not eradicate it from the world, it became important to look at all the species, and therefore Plasmodium ovale is now becoming recognized as a species of potential importance with respect to the elimination programs that are there. Now, with these studies, other people and ourselves have noticed that when they looked at other genes, these are Plasmodium ovale genes that are expressed on the eukaryote surface. There were also 2 different populations. They're also split into 2 different populations. The polymorphisms were there. There were 2 types. These genes were looked at, and then in 2004, cytochrome B, then cysteine protease, and then ourselves looked at dihydrofolate reductase. Colin Sutherland looked at 2 different reticulocyte binding protein 2 and other proteins. And we got together with Colin Sutherland to suggest, after seeing all of this, that there actually were probably 2 different populations of non-recombinant Plasmodium ovale. Why do we say non-recombinant? Because we never found a Plasmodium ovale that had, let's say, the polymorphic type of one classic, but the polymorphic type of a variant in the same parasite population. So there were always 2 separate populations that were found. Therefore, they coexist throughout West Africa, throughout East Africa, and even in Papua New Guinea, in other places where P. ovale is there, but they've never seen a recombinant form of the 2 parasites. And therefore, Colin Sutherland proposed that we should call one of them the classic one, to give it the name Plasmodium ovale curtisi, and the variant one, Plasmodium ovale Wallikeri. Now, Chris Curtis and David Walliker are both eminent malariologists who have sadly died now, and the names were given in their honor. We proposed in this article, which we published in 2010, that they should be considered as two species, although we were only relying on about 5 or 6 genes. With time, the sequence, the whole genome sequence has been obtained from the two parasite populations, the classic, the variant, the Cortesi and the Wallikeri ones, as it's become known. And it confirmed that there were indeed, throughout the genome, there are differences that are sufficient to tell them apart. And they are so different, they are as different as two species as, let's say, Plasmodium vivax is from Plasmodium falciparum. There are big differences, and therefore this has confirmed that we are indeed dealing with two species. Now, the mistake that has been made, and I'm partly guilty of this, is that Plasmodium ovale Cortesi, the way it is done here, and ovale Wallikeri is done here, is written as Plasmodium ovale and Cortesi. This is called a trinomen. It's 3 names for the things. And by the rules of the International Code of Zoological Nomenclature, a trinomen must denote a subspecies. And therefore, this has caused the confusion in the community, because out of all the papers that are published, a number of authors consider them as species, and a number of other authors say that they are subspecies. It is important to know which it is for any future studies on these things. And indeed, some of the people who did call them species in one paper would call them subspecies in another paper. A couple of months ago, I got together with colleagues in Japan and in Edinburgh to correct this mistake, and we published a paper which has appeared in Trends in Parasitology a couple of weeks ago, in which we proposed that the 2 species of P. ovale that were formerly known as P. ovale should be now called P. ovale Cortesi. We just put the 2 names together, and P. ovale Wallikeri. Within about a week of our publishing, we had a clash with the international. We had a complaint, an objection that is going to be published soon, saying that this is not an allowed thing to do, because we should keep the name P. ovale for one of them and then name the other one a new name. But we are now resisting this, and we are objecting to the objections. And we hope that these 2 names will indeed be kept in future as to denote these 2 species of Plasmodium ovale. Now, what are the future investigations that will be interesting to do? Now we can tell the 2 species apart, provided we do molecular analysis, because it is still impossible to tell them apart morphologically. We don't understand whether there are indeed any biological differences between these 2 species. From their published data, and as there's not much published data, the data does not allow to show that there are any serious differences in the way the course of infection is. There's some indication that the relapse pattern might be different, some indications that the fever might be higher in one than in the other. But these are really, these are really very weak data on which to base any conclusion. The other interesting thing is, from a biological point of view, is how did they evolve? How can 2 species evolve in the same host with the same mosquitoes that transmit them to become 2 separate species that do not recombine together when they are so close together? In a sense, they are very similar. They occupy the same niche, and this is quite an interesting subject for people who are interested in evolutionary biology. I would like to end this talk by saying, keep an open mind. Many, many decades after discovering and studying these parasites, we were surprised to discover that there were 2 species. And it's the molecular aspect of it which has made it possible to tell the 2 species apart. And it is possible that other parasites, including Plasmodium falciparum and vivax, might eventually we find that there are differences that have been not observed biologically and might have a clinical implication. Therefore, one should always keep an open mind when studying biology, even if there is a lot of publications on it, and that is an important message that I would like to give today. Thank you for your attention, and I'm happy to answer any questions.
Thank you, Prof. Georges. It was very insightful and as well as interesting. So can we have the member of the audience to ask any question? You may type out your question in the chat box, Q&A chat box, and I will then read it out to Prof. Georges to answer.
Stop the share. There you go.
Okay, Prof. Georges, we have a question from Prof. Fong. So he has asked, what about Plasmodium cynomolgi?
What about Plasmodium cynomolgi, Prof. Fong? Good morning, Prof. Fong. Plasmodium cynomolgi, whether it also has different subspecies or different species, is that the question, Prof?
I assume that's the question. Yeah, he said yes.
There are two subspecies of Plasmodium cynomolgi. There's one called cynomolgi, and the other one discovered in if I'm right, 1959 by Professor Garnham, which he called Plasmodium cynomolgi bastianelli. But the two species, it is not known whether these two species recombine together. Nobody has attempted, as far as I know, to recombine them. And it is possible that they are not two subspecies, but they're two separate species. And with the studies that we are doing now and the strains that are available, it might be possible that some other parasites that are very morphologically similar to P. cynomolgi are indeed like Plasmodium ovale, cryptic species that coexist into the monkey. I mean, one has to say that with respect to the parasites in the macaques, in a period of 5 years in the early 1960s, American and Malaysian researchers working at the Institute of Medical Research in Kuala Lumpur uncovered 5 new species of parasites in 5 or 6 new species of parasites in macaques and gibbons in Malaysia. And then they stopped the research. And it is possible today that there are other species there that we don't know about. The difficulty nowadays, of course, is sampling primates, is non-human primates, is not very easy, as Prof. Fong knows.
Thank you, Prof. Georges. I think that would have answered Prof. Fong's question. We have another question from Professor Lau Yiling. So how many genes are needed to distinguish between two species, or genome sequencing remains the sole alternative?
Well, in order to really confirm a big difference, the more genes you have, the better it is. But ultimately, a couple of genes will be enough to distinguish between two species. I mean, it is easy to distinguish between using, let's say, the gene MSP1. If you know the sequence of MSP1, we know the sequence of that in P. vivax, P. malayi, P. falciparum, and P. ovale. And just by looking at that gene, one can the same for the small subunit ribosomal RNA. However, in order to confirm that there are indeed two species that are genetically very separated, the more genes you have, the better will be a phylogenetic tree constructed. It is possible that two species will have some genes that are very close, similar together because they happen to be conserved. And if you start using these genes first, then you might not realize that two species are separate and other genes are quite different. And therefore, the reason why we decided to correct the nomenclature now is because now with the genome sequence, it is not only 4 or 5 or 6 genes that we found that they are dimorphic, it is the whole genome is overall dimorphic. And therefore, the more genes you have, the better you have, the more robust your argument is to call two morphologically identical species different. And that's the difficulty in this, is that in protozoology, often parasites, protozoans look very similar, but they are different organisms.
Thank you, Prof. Georges. I think that would have answered Prof. Lau's question. Any more questions from the audience, the participants? So I have one question, Prof. Georges, about the these parasites forming species, subspecies. What do you, what could be the driving factor? Is it the host factor or the resistance or treatment, or what do you think could potentially be the reason?
For the evolution of two separate species?
Yes, yes.
Well, I don't think it's treatment because we've only started treating malaria in the last, shall we call it, since quinine was discovered. Quinine was discovered in 1640s. Of course, it hasn't been used extensively in Africa at the time, but let's say it's been used in Africa since the 1880s onwards, and that's not that much time to drive, I think, two species to evolve. So it is not treatment. And all the studies that have been done on P. ovale observations, if you treat them, they all respond exactly the same. So there isn't one that doesn't respond another. We don't really know very much about which vectors transmit them. They can be transmitted, we assume that they can be transmitted by the same vectors, but we're not sure. Because the studies that have been done, the experimental studies that have been done in the 1930s and 40s and 50s. And these were done on samples that were considered to be P. ovale. And we don't know whether they were P. ovale of type Curtisi or the P. ovale walliceri. But whenever people tried to transmit, they did succeed in transmitting using different mosquitoes. But it is very rare to have somebody who's used two parasites. We don't know of anyone who's used two parasites which are known to be different molecularly and have tried to transmit them by the same anopheline. It is possible that if some parasites, because we know that some anophelines will transmit some strains or some species of Plasmodium, but not others. And therefore, it is possible that if you look at evolutionary speaking, during, let's say, the last 500,000 years, that P. ovale was in humans, or indeed maybe P. ovale was in apes, in gorillas or chimpanzees before. It is possible that in Africa, if we assume that the origin is in Africa, which is probable, but nobody can prove this for sure. But it is possible that if the mosquitoes that were feeding on the host were different mosquitoes, and we know there are many species of anophelines in Africa, if one of them transmitted one and not the other, then the two will never recombine. And therefore, eventually they will speciate because, well, species, when you kind of isolate them, will become different and different. We go back to Darwin's finches. So unfortunately, the only way to do that now is to experimentally infect people because we can't grow them in culture, and we can't, the non-human primates that are used experimentally are not susceptible to P. ovale. And therefore, we'll have to start infecting humans again, which is not easy.
Thank you, Prof. Thank you.
Pleasure.
We take one last question before we end this
Sure.
session from Sarah Ahmad. How
Yeah.
could you envision clinicians could use this phenomenon to prevent or treat the clinical presentation?
Well, for the time being, irrespective of which species has been discovered in a person, once people have done the molecular analysis, the treatment that is used, mainly chloroquine plus bromoquine, works very well. They haven't, fortunately, well, we haven't seen any differences now, at least with respect to clinical presentation, any major differences, or with response to treatment. Therefore, I think that from a clinical point of view, it is probably unimportant. Now, from an epidemiological point of view, it'll be interesting to see whether some infections with one relapses more than the other, or whether the parasites that stay in the liver as hypnozoites have a much longer or shorter period in which before they get activated again, and whether the other important thing is that in areas where you're removing Plasmodium falciparum, let's say in West Africa, because treatment of ACTs is becoming common, is it likely that all the P. ovales that were, in a sense, sub-patent before because they were undiscovered they might have been suppressed. They might have been suppressed by Plasmodium. Will now we have a surge in Plasmodium ovale that comes up? Although I say that Plasmodium ovale is relatively rare to have severe symptoms, they do occur sometimes. And as anybody who's ever had malaria from P. ovale, and I've known people who had P. ovale, it is a very unpleasant disease. So this is not a disease that is mild in the sense that it's like a kind of a passing flu where you still go to work but you sniffle. You're in bed and feeling really bad. Therefore, it does cause morbidity, and therefore. If the two parasites differ in their morbidity, and we don't know whether they do, that might be important in the epidemiology. That might be important from elimination programs, but they clearly have to be taken into account, whereas they were completely ignored before, like P. vivax was for many years.
Thank you, Prof. I think that would have answered Sarah Ahmad. So we have come to the end of the session for today, Breakfast at UMHealth. Thank you, Prof. Georges Snounou, for your time and staying up
very
late,
much.
1
It's been a
a.m.
pleasure.
Right? So thanks a lot, and thank you for the participants. And now I'll pass it over to Prof. Sanjay to moderate the next session. Thank you, Prof. Georges.
Thank you very much.